Good morning, students.
Good morning, madam.
How are you this morning?
Thank you, madam.
Okay. Today we'll be learning a new topic, which is genetic engineering and population genetics. First of all, we'll be moving on to our motivational activity, which is word search activity. Okay, there are some words that you have to find out on the table given, and it's a 2 minutes activity. Okay, and it's 10 marks each. Okay, I hope that you students will try to answer those questions um, on the time limit that is given. Okay, these are the worksheets. The questions are in the worksheets. Okay, it's a 2 minutes activity. You may start. Okay, I know that you have completed those activity.
Yes, madam.
Okay, uh, there are 10 words. Okay, the first one is DNA, the second one is genetics, third one is mutation, the fourth one is heterozygous, the fifth one is homozygous, the sixth one is allele, and the seventh one is gene. The eighth one is migration, the ninth one is chromosome, and the 10th one is dominant. Okay, the solutions um, the solutions are here. Those are the solutions, okay, for the exercise, the motivational activity that was being provided. Okay, um, those are some of the words that you'll be seeing on this topic. Okay, those are the words that are mostly used in genetic engineering and population genetics. Okay, we'll be moving on to our lesson notes. Okay, I'll be introducing the topic. The topic is genetic engineering and population genetics. Okay, those are the two topics that will be covered on this 1 hour lesson this morning. Okay, I hope that you students will cope up with me during my explanation of the lesson notes. Okay, these are the handout of the lesson notes. You may have one each so that you can cope up with me and follow what is being taught during this lesson. Okay, there are 3 learning objectives in this lesson. Okay, the first learning objective is you have to define what is genetic engineering. Okay, the second objective is you have to explain the role of population in evolution. Okay, and the third objective is you have to calculate the percentage and frequency of population by using the Hardy-Weinberg principle. Okay, you have to, those are the 3 learning objectives that you have to meet, or you have to meet those learning objectives at the end of the class. Okay, at the end of this 1 hour class. Okay, moving on. Okay, can anyone tell me what is genetic engineering? Can anyone explain what is genetic engineering or recombinant DNA? Okay, I think so. No one can answer that question. Okay, genetic engineering or recombinant DNA refers to the different manipulation of DNA or genome to alter organisms' characteristics, phenotype in a particular way. Okay. Another name of genetic engineering is recombinant DNA. Okay, we know that DNA is the deoxyribonucleic acid. Okay, that's DNA. Okay, we have DNA in our gene. Okay, um, that's DNA. Okay, refers to the different manipulating of DNA or genome. Genome is a gene. Okay, it's genome is gene. Okay, to alter organisms' characteristics, phenotype in a particular way. Phenotype is a physical characteristics of an organism. Okay, that's phenotype. Okay, moving on. This means changing base pair, deleting whole region of DNA, or introducing an additional copy of a gene. Okay, changing base pair. We know there are 4 base pair. Okay, uh, adenine, uh, glycine, thymine, and guanine. Those are the base pair. Nitrogen base pair. Nitrogenous base pair that is present in the chromosome. Okay, uh, deleting the whole region of the DNA or introducing an additional copy of a gene. That means what genetic engineering does. Okay, the DNA of an organism that is altered by incorporation of gene is known as the recombinant DNA. Okay, the DNA of an organism that is altered by incorporation of gene is known as recombinant DNA. The replication of altered DNA is known as cloning. Okay, all of us know what is replication. Okay, replication when a DNA strand is being when a DNA strand is being broken out. Okay, when a double helix is being broken out or separate, that means replication is taking place. Okay, um, moving on. Recombinant DNA, okay, is something referred as the chimera DNA since it involves combining 2 or more strands of DNA from 2 different organisms in order to create a new strand of DNA. Okay, it separates. Okay, so that it separates. After that, the nitrogen bases, they form another new strand. Okay, to form the complementary base pairs. They pair up and they form another new strand from a old strand. Okay, um, genetic engineering utilizes recombinant DNA. Okay, genetic engineering, it uses up recombinant DNA. Okay, the important tool for recombinant DNA, restriction endonuclease. Okay, this enzyme cut DNA molecules at specific sites. Some restriction endonuclease cleave both strands of DNA simply at the same point within the recognition sequence. As a result of this type of cleavage, the DNA fragments with blunt ends are generated. Okay, that's the enzyme that cuts the DNA molecules. Okay, it is known as the restriction endonuclease. Okay, the exonucleases is an enzyme that removes nucleotides from the ends of the nucleic acid molecules. The exo, it removes the nucleotides. Okay, it removes totally the nucleotides. The DNA ligase, it joins 2 fragments of DNA by synthesizing the phosphodiester bond. The DNA polymerase, it synthesizes a new complementary DNA strand of an existing DNA or RNA template. Reverse transcriptase is also an important type of DNA strand called the complementary DNA. The vectors, DNA molecule used as a vehicle to artificially carry foreign genetic material into another cell where it can be replicated or expressed. Okay, host organisms, bacteria, E. coli is widely used as a host in the recombinant DNA. Okay, E. coli is a material that is being used in the genetic engineering to take up the role of as of a host organism. Okay. Another one is foreign DNA. The desired DNA segment, which is to be cloned, is called as DNA insert or a foreign DNA. Okay, those are the materials that is been cloned during this during recombinant uh, during the recombinant transferred DNA. Those steps, this one is used to this material is used to be cloned. Okay, and transgenic organism, it carries recombinant DNA, example, bacteria. Okay. Moving on to the steps in recombinant DNA technology. Okay. Selection and isolation of DNA insert. That's the first step. Okay. We have to isolate the DNA. Okay. We have to select and isolate the DNA. Okay. Selection of a DNA segment of interest, which is to be cloned. Okay. Then the second step is the selection of the suitable cloning vector. Okay. A suitable cloning vector is selected in the next step of recombinant DNA technology. That's the second step. Okay. The third step is introduction of DNA cloning vector to form recombinant DNA molecule. The DNA which has been extracted and cleaved enzymatically by the selective restriction endonuclease enzymes are ligated, joined. Ligated means it joined by the enzyme ligase to vector DNA to form the rDNA molecule. Okay, that's the third step. The fourth step is recombinant DNA molecule is introduced into a suitable host. Okay, suitable host cells are selected in the rDNA molecule form is introduced into the host cell. The process of entry of rDNA into the host cell is called transformation. Okay, transformation. Usually selected hosts are bacterial cells like E. coli. However, yeast fungi may be utilized. Okay. The fifth step is selection of transformed host cell. Are those host cells which have taken up the rDNA molecule. Okay. The transformed cell separated from the non-transformed cell by using various methods, making use of marker genes. Okay. Expression and multiplication of DNA insert the host. That's the last step, which is gene cloning. Okay. Finally, is to be ensured that the foreign DNA inserted to the vector DNA is expressing the desired character in the host cells. Also, the transformed host cells are multiplied to obtain sufficient number of copies if needed. Such genes may also be transferred and expressed into another organisms. Okay, those steps I have simplified it by putting it on the chart. Okay, the steps in recombinant DNA technology. This is the diagram, and this is the summarized steps of what I've already read out. Okay, from the lesson notes. Okay, these are the host. Okay, the host is being taken out. Okay, the plasmid. This is a human cell DNA. Okay, it's been taken out. Okay, the gene. It's been taken out. Okay, then this part, the one that's been cut, it is been cut by that enzyme. Okay, the restriction enzyme. Okay, they cut this end. Okay, it's known as the sticky ends. Okay, and moving on. Okay, those sticky ends, it covers or it joins. Okay, with the plasmid. This is the plasmid. Okay, then after that moves on. Okay, this gene X is being inserted here. Okay, to the plasmid. Okay, the recombinant DNA plasmid. Okay, then it moves on. Okay, then this is the cell. Okay, and both of it is been present inside. Okay, this one is been present with, this is the bacteria. Okay, the E. coli bacteria. It acts as a host. Okay, then after that it multiplies. Okay, it produces many, okay, many plasmid and bacterial cell. Okay, it multiplies, it reproduces, okay, to form many, okay, many cells. Okay, bacterial clone carry many copies of the human gene. The first step, we have to isolate DNA from the two sources. Okay, this is the two sources. We have to isolate the DNA from there. Then after that, we have to cut both DNAs with the same restriction enzyme. So we have to cut this. Okay, cut the DNA by using that enzyme, which is the grid restriction enzyme. Okay, then the third one is mix the DNAs, they join by base pairing. Okay, then we have to mix them, join them. Okay, then the fourth step is add DNA ligase to seal the DNA with covalent bonds. This is the DNA ligase which seal that open part, okay, in the DNA. Okay, then the fifth part is put plasmid into bacterium by transformation. So this is the bacterium. Okay, it's been put together with the plasmid. Okay, then after that, clone the bacterium. So the bacterium has been cloned to form these many cells. Okay, it reproduces and it multiply. Okay, that's all the explanation of the steps in recombinant DNA technology. Okay, the moving on to the next part, the advantage of genetic modified organism. Genetically modified organism refer to the genetic material that has been artificially altered through genetic engineering to change its characteristics. Okay, other advantage includes insect resistance, okay, high yield, decreased use of pesticides, and less deforestation. Okay, disadvantage of GMO, which is not 100% environmentally friendly, food supply at risk, new diseases coming through, economic concerns, and ethical issues. Okay, those are the advantages and disadvantages of genetically modified organism. Okay, DNA cloning is a process of making cloning techniques are used for many purposes in molecular biology, biopharmaceutical, gene therapy, gene analysis. Okay, the application of recombinant DNA technology, production of transgenic plants and animals, production of hormones, vaccines, and biofuels, production and diagnosis of diseases. Danger of genetic engineering, spread of new diseases, effect on evolution. Nature has provided several barriers for exchange of DNA between prokaryotes and eukaryotes. Okay. Recombinant DNA technology permits exchange of DNA between these two classes of organisms and thus interferes with the natural process of evolution. Okay. Population genetics refers to the study of distribution and changes of allele frequency and interaction of allele in a population. Okay, that's population genetics. A population is affected to the form and evolutionary forces, namely natural selection, genetic drift, and mutation. Okay, those are the affected that always affect the population. Okay, those are the forces, and the gene flow, migration. Okay? The smaller a population, the more susceptible it is mechanism like natural selection. Population is defined as a group of individuals of particular species occupying a definite space in which the organisms interact, interbreed, exchange genetic materials. Okay, that means a population. Okay, a population only same kind of animals, the same kind of animals, they stay together. Okay, like a family, or stay together as one family. That means population. Okay, they interbreed. Okay, they exchange genetic materials and they interact with each other. Okay, Role of population in evolution. For the processes of evolution to occur in the system, there should be changes in the genetic equilibrium. Gene pool is the sum of total genes of an individual in a population. A gene pool of population describes gene present in the population, proportions of different kinds of genes, and pattern of distribution of genes in the individual of population. Okay, a population and a population with a gene pool. Okay, allele frequency represent the frequency of a gene allele variation in a population. Okay, allele means gene. Okay, the different gene that a organism has, that's known as the allele. Okay, alleles are position or genetic locus on a chromosome. Okay, allele in a chromosome, okay, like it has the gene in a chromosome. There are different kinds of genes. Okay, there are many different kinds of genes that are present in a chromosome. Okay, position or genetic locus on a chromosome. Okay, changes in allele frequency over time can indicate that genetic drift is occurring or that new mutations have been introduced into the population. Okay, population. Okay, sometimes they migrate. Okay, they migrate to another area. Okay, so when they migrate to another area, they taking that gene. Okay, when they migrate, okay, they may be interbreed or interact with other animals that has different gene. Okay, that has another kind of gene. So they interbreed or they interact with each other, and therefore the population from where he left, okay, it will decrease. Okay, the gene frequency or the genome will decrease. Okay, that simply means that statement. Okay, changes in the allele frequency over time can indicate that genetic drift is occurring or that new mutations have been introduced into the population. Okay, allele frequency, genotype frequency refers to the total number of kind of individual from a population, all of which exhibit the similar character. Okay, that's genotype frequency. The Hardy-Weinberg principle, okay, also referred to as Hardy-Weinberg equilibrium. It is the fundamental concept in population genetics, a mathematical equation describing the distribution expression of allele forms of a gene in a population, and it expresses conditions under which allele frequency is expected to change. Okay, the Hardy-Weinberg principle, it has a formula. Okay, it has a formula. The formula is p squared plus 2pq plus q squared is equal to 1. Okay, here it's been written. I wrote it on the chart so that you can see. Okay, when I'm explaining. Okay, population Hardy-Weinberg equation. P squared plus 2pq plus q squared will give you is equals to 1. Okay, this whole formula is equals to 1. Okay, p squared means the frequency of homozygous dominant genotype. Okay, homozygous means it's the same. Okay, that's homozygous. Dominant means it's dominant over, like for example, a white butterfly is more dominant to the black butterfly. Okay, so dominant genotype, it will be written as dominant, it will be capital. Okay, it will be a capital letter. Okay, genotype means the things that we can see. Okay, it's gene, right? We can see the gene in our body. That's genotype. Okay, so B is the frequency of homozygous dominant genotype. So it's B. Okay, capital letter BB. That's dominant. Okay, 2PQ, frequency of heterozygous genotype. Heterozygous means two different allele. Two different allele. That's heterozygous genotype. So heterozygous, it will be one capital B and one small b. That's heterozygous, right? Heterozygous genotype. One capital B and one small b from two different alleles. Homozygous is the same, from same allele. That's homozygous. Okay? And Q squared is equal to frequency of homozygous recessive genotype. Recessive means two small b. Okay? That's recessive. It's not known. That means recessive. Okay? Too small b. So that's the explanation of this whole formula. Okay? P squared plus 2PQ plus Q squared will give you 1. Okay? I hope that you have understood that explanation. Isn't it, student? Have you
Yes, madam.
understood? Okay. That's the explanation of Hardy-Weinberg equation. Okay, we'll be moving on to the assumptions of Hardy-Weinberg principles. The assumptions is the only true of a population is in equilibrium, and the following conditions are true. Okay, generation must not overlap. Okay, those are the assumptions of Hardy-Weinberg. Okay, generation must not overlap. No natural selection, mutation are negligible. No gene flow. Students, this is the continuation of my teaching, which is the assumption of Hardy-Weinberg principle. Okay, moving on to the fourth one, which is no gene flow between population, migrate in and out of the population. Population is larger, no genetic drift. Individuals are mating randomly. Okay, moving on to the example. There are one example. Okay, you may see that. Okay, this example of a question is given, which is within a population of butterflies, the brown wing color, which is B. So B, capital B, you know that's dominant. Okay, is dominant over the white wing color. Okay, so white wing is recessive. Okay, so 36% of all butterflies are white. Okay, 36%. And using the information, calculate the following. We have to calculate the percentage butterflies in the population that is heterozygous. We know heterozygous is 2PQ, which is this. Okay, heterozygous will be a capital B and a small b is equals to 2PQ. Okay, white is recessive, means 2 small b. Okay, and it's 36% of butterflies are white. Okay, therefore we have to calculate that. Okay, that 36% over 100, which will give you 0.36. Okay, and Q is equals to 0.6. So if Q is 0.6, then P is equals to 1 minus 0.6. Okay, this one. Okay, 1 minus 0.6, the answer is 0.4. Okay, 0.4, and 2PQ is 2, then you'll multiply them. So 2, bracket 0.4, bracket multiply by 0.6 will give you 0.48. Then you multiply that 0.48 multiplied by 100 to give you the percentage of the butterflies in the population. Okay, and part B is to find the frequency of homozygous dominant individual. Okay, homozygous dominant, which is 2 capital B. Okay, same, and the formula is P squared. This is the frequency of homozygous dominant. So P is 0.4 squared will give you 0.16. That's the solution of that example. Okay, moving on, the factors that affect genetic equilibrium. Factors that affect is mutation, recombinant during sexual reproduction, and genetic drift. Okay, those are the factors of genetic equilibrium. Okay, uh, the diagram of founder effect and the diagram of bottleneck event. Okay, gene migration, immigration results in addition of new alleles in the existing gene pool and changes the allele frequency. Okay, that's all for our lesson notes this morning. Okay, I'll be moving on to the group activity work. Okay, the group activity work, I'll divide yourself students into 3 groups. Okay, there are some questions that has to be answered. Okay, it's 3 marks questions. Okay, 3 marks each. Here are the worksheet. You have to answer it. It's just a 5 minutes activity, group work activity, and it's just 3 questions. Okay, 3 groups. Okay, answer those questions, then we'll mark it at the end of this activity. You may start. It's a 5 minutes activity. You may discuss. Okay, all students has to be involved in those discussion of those 3 questions. Then we'll discuss it together in class. Okay, only 5 minutes. I know that you have completed those activity. Okay, I know that you all finished.
Yes, madam.
Okay, we'll discuss those questions. Okay, the first question is 1 mark. Okay, the first question is write the advantage of genetically modified organisms. Okay, the answer is, for that question, genetically modified organism refer to the genetic material that has been artificially altered through genetic engineering to change its characteristics. If you have that answer, place a tick and put 1 mark. Okay. The second question is write 4 advantages of genetically modified organisms. Okay, it's a 2 marks question. The first one is insect recessive, high yield, decreased use of pesticides, less deforestation. If you have that answer, mark it right, place 2 marks in that question. Okay, the third question is what 4 disadvantages of genetically modified organisms. Okay, the first one is not 100% environmentally friendly, food supply at risk, new diseases, economic concerns, and ethical issue. If you have that answer, place a tick. It's 2 marks. Okay, the total mark of that 3 questions is 5. Okay, moving on to the quiz. Okay, moving on to the quiz question. Okay, explain the steps in recombinant DNA technology. Okay, it's a individual work. Okay, you have to, this is the paper, you have to write your answer in this paper. Okay, provide or write all the steps in recombinant DNA technology. Okay, you write the steps, then I'll ask individual students to answer. Each one of you to answer this. Give me 1 step each. Okay, it's only 2 minutes. You have to do that, answer that question for the interval of 2 minutes. You may start. Okay, the steps. Okay, first step is the selection and isolation of DNA insert. The second step is selection of suitable cloning vector. The third step is introduction of DNA cloning vector to form recombinant DNA molecule. The fourth step is recombinant DNA molecule is introduced into the suitable host. The fifth step is selection of transform host cells. And the sixth step is expression and multiplication of DNA insert in the host gene cloning. If you have that answer, mark it right. Okay, that's the quiz questions. Okay, moving on to our video. I have a educational video with me for you to watch. Okay, and you can have a clear knowledge of this this lesson. The first video is based on the genetic engineering.
Many years ago, diabetics who needed insulin to survive had to inject cow or pig insulin into their bodies. This was not ideal for many reasons. Today, the insulin that diabetics inject is human insulin, and this is produced by microorganisms such as the E. coli bacterium and certain strains of yeast. But how do these microorganisms produce human insulin? Genetic engineering. Scientists are able to turn certain microorganisms into mini factories that make useful substances that improve our health, the environment, and the economy. But what is genetic engineering? An organism's DNA makes up its genes, which code for all the proteins the organism needs to survive. Each gene codes for a different protein, or part of a protein. Genetic engineering is the manipulation or changing of the DNA of an organism. Genetic engineering involves removing a gene from one organism, which is called the donor, and transferring it to another organism, which is referred to as the recipient. The recipient is called a transgenic organism, or a genetically modified organism. There are two basic purposes of genetic engineering. Sometimes, we require large volumes of a particular protein to be made. Scientists use transgenic microorganisms to produce large volumes of this protein. For example, insulin, growth hormone, or vaccines. An organism, B, can have a gene from a different organism, A, introduced to give it, B, some advantage that A has naturally. Organisms like B are called genetically modified or transgenic organisms. For example, a gene that manufactures a toxic chemical in a bacterium can be introduced into a maize plant to make the plant toxic to certain insects that would eat it, such as caterpillars. Let's now look at the process of genetic engineering. How do you transfer a genetic characteristic from one organism to another? Although it's a very complex process, it can be summarized rather simply. We'll use the example of how the insulin gene is inserted into a bacterium in order to manufacture large amounts of human insulin. The gene that codes for human insulin is found on chromosome 11 at position 15.5. The insulin gene is cut from chromosome 11 using special enzymes called restriction enzymes. Bacteria possesses small circular strands of DNA floating in their cytoplasm. These strands of DNA are called plasmids. A plasmid is extracted from the bacterium cell. The plasmid is cut open using the same restriction enzymes that were used to cut the insulin gene from the human chromosome. The restriction enzymes leave sticky ends, where one of the two DNA strands is slightly longer than the other. Because the same restriction enzyme was used to cut both the human DNA and the bacterial plasmid, the sticky ends are complementary and will allow for joining using complementary base pairing. The insulin gene is joined to the plasmid by the sticky ends. An enzyme called ligase is used to complete the joining of the two pieces of DNA. The modified plasmid is then reinserted into the bacterial cell. The modified bacterium is a genetically modified transgenic organism because it contains some human DNA along with its own bacterial DNA. Its bacterial DNA is called recombinant DNA. Bacterial DNA recombined with human DNA. The bacterial cell is placed in a fermenter which allows for rapid asexual reproduction in ideal conditions with the optimal temperature, pH, and lots of food. Because the bacteria reproduce asexually, the offspring are all clones of the original transgenic bacterium. They all have identical recombinant DNA. The bacteria express their own genes in order to survive. In other words, they make their normal bacterial proteins. But they also express the human insulin gene and make human insulin. The fermenter has become a culture of mini factories, all producing human insulin. The insulin is then extracted, purified, and packaged. Humans can now inject themselves with human insulin. And that is how genetic engineering works.
If you like the video, give it a thumbs up.
Okay, students, that is a short video on genetic engineering. I know that you understand, okay, the whole lesson that has been taught. Okay, moving on to the recapping of the notes. Okay, these are the chart, okay, that has been provided. Okay, it's been simplified, the steps in recombination DNA technology. Okay, and the population genetics, the Hadwiin-Beck equation, its explanation. Okay, and you have to know the main concept that you have to know in this lesson is to know the steps of this recombinant DNA technology in this topic of genetic engineering and the Hadwiin-Beck equation. Okay. Okay, students, that's the summarized notes. Okay, and we'll be moving on to the homework. Okay, I'll be giving out homework questions for you to answer. Okay, then we'll be discussing it on our next class. Okay, those homework questions. Okay, here are the worksheet of the homework questions. Okay, and my teaching aids are chart, the educational videos, and the lesson notes that has been put up on Microsoft Word. Okay, okay, students, you may that's the end of our class. You may set up the desk. Okay, the table, the chairs, pick up the rubbish. Okay, that's lying on the floor, and take out your books, and you wait for your next class. Thank you.